Good evening, everyone. We're just going to give it a minute before we start off to let people join. All right, so first of all, I'd like to say thank you so much for joining us this evening. We're going to be talking about the painless restoration of children's teeth. My name is Abdul Javid, and I'll be the host for today's meeting. I'm a second-year medical student studying here at UCL, and I'm very pleased to be hosting this lecture, which will be very informative. So in today's lecture, Professor Anne Young and Professor Paul Ashley will be talking about the progress made in the painless restoration of children's teeth and the journey that it has undertaken in recent years to get these new materials into clinical trials. So in order to keep these lectures as interactive as possible for the audience, we're going to be using the Q&A answer feature on Zoom. So please, as the lecture progresses, just any questions you have in mind, submit them into the Q&A and then they will be answered at the end. So the hashtag to use on Twitter for this event is hashtag FMS Lectures, and this will be recorded, and the link to the recording will be sent out via email. So just to make sure that you guys are aware of that. So before I introduce our speaker, we would like to know about our audience a little bit. So we're going to be sending out a poll soon about the type of group you belong to in terms of whether you are a prospective student and their families, undergraduate students, PGT students, PGR, PhD, just in general academics, patient carer, or any other. So I'll give you guys a few seconds to fill that in first. All right, so it seems that there's a wide variety of people, and we'll see how many will have attended today in a bit once the poll ends. So yeah, most people are prospective students and their families who I guess are looking to study in a similar field of study. We've got a few undergraduate students, a few PGT, no PGR, a PhD, some academics, and a few others. That's great. It's good to know that we have a wide variety. So thank you so much to everyone that voted. It really helped. And I'm delighted to now introduce our speakers. So first of all, we have Professor Anne Young. And Anne Young is a professor of biomaterials at the Eastman Dental Institute, working on new materials for tooth and bone repair. So the smart composites that her group has been developing in collaboration with Scotland the Dental Company are designed to enable the restoration of children's teeth without the need for anesthetic or drilling. So moving on to Professor Paul Ashley. Professor Paul Ashley has been working in the UCL Eastman Dental Institute since 1998, where he now runs the professional doctorate in pediatric dentistry and is deputy program director for the MSc in pediatric dentistry. Professor Ashley also works closely with Professor Anne Young, supporting her work on the development of novel materials for dental restoration, and he has an interest in evidence-based dentistry and the management of pain and anxiety in children, which is something that's very important. I'm sure we can all agree on. So thank you, Anne and Paul, for joining us today. I'm going to give it over to you guys. Take over.
Okay, thank you. Thank you to UCL for the invitation. Thank you to everyone who's tuned in, and thank you to the introduction as well. So I'm going to set the scene as the kind of clinical part of this partnership with Professor Young. I guess my role was to try and explain to her what the problem was. And then Anne's going to talk to you a little bit and dive into the chemistry of the material that she's developed. And then we'll finish the talk by just unpicking or explaining a little bit how we go from the laboratory to the clinic, because that is as complicated as, frankly, developing the material has. So we've got about half an hour to speak. We're actually compressing, I think, probably between 15 to 20 years of work in the half an hour. So I'm sure we'll skip over lots of really important bits, but hopefully you get a flavour for what we're doing. So let's start off, like I say, with setting the scene. And the problem that we're trying to manage is really dental caries. And dental caries, it's not as, I guess, as interesting or as talked about as diabetes or cardiac disease or other things, but actually it's the most prevalent, it's the most common non-communicable disease worldwide. Affects children and adults, but we're particularly interested in children here. About 40% of 2 to 11-year-olds have got dental caries, and about 60% of 12 to 19-year-olds have got dental caries. 5 to 10% of global healthcare expenditures. That's a huge amount of money. And it's an even bigger tragedy because we know how to prevent caries. We know what causes it. We know how to stop it. But that's not what today's talk is about. because even though we know how to stop caries, it still happens. And it tends to be, particularly in children, largely untreated. About 45% of people with caries around the world, it's not treated. So why is this? And if I can have the next slide. So we're going to think really about children here, and the reasons why caries in children is untreated. Like all problems, it's complex, it's multifactorial. Sometimes it's around cost or funding models. There's simply not enough money given to manage the treatment of children. Sometimes it's around access. So if you look how dental care, how dentists are distributed across the population of the world, about 69% of the dentists, only 27% of the population, because at the end of the day, the things that make money, that allow somebody who is working to make a profit, tend to lead more towards the cosmetic end. That tends to be where the money is. Capability, skill mix, treating children is a little bit more difficult than treating adults, and often you need some extra skills to be able to do that, which is really my other part of my job, is trying to skill people up. But the big headline reasons why caries in children tends to remain or is largely untreated are partly because the existing dental materials we have aren't really great, they aren't really good at managing or filling children's teeth, and also, rather obviously, children are difficult to treat. Adults at the best of times don't want to have fillings done, don't want dental injections, don't want to have their teeth drilled onto. And children are obviously even more complex to manage in that regard, and are more likely to not want to cooperate. So next slide, please. Thank you. So what can we currently use to fill children's teeth? What options are available? And up until about sort of 10, 20 years ago, dental amalgam, mercury-based filling material was the standard. Not pretty, but very straightforward to use, really tolerant to moisture, which obviously is a big problem in the mouth, and really was the mainstay. It's effectively banned now, amalgam, not so much because of the mercury being released into your body, but more because just handling it, just disposing of unused amalgam is really complicated. So that's gone. So the next common option we use are glass ionomer cements. I'm not going to go into detail as to what the chemistry of them is, but suffice to say that they are tooth-coloured. They're relatively straightforward to place, but they're not that strong, certainly not compared to amalgam, and certainly not compared to the material we use most often. Next slide, please, Anne. Which are dental composites. So dental composites are probably the most commonly used filling materials nowadays. If you've had fillings at your dentist, it's likely you've had dental composite placed. They're tooth-coloured. They are stronger than glass ionomers. On the face of it, you'd have thought they were a nice option for children, but they have lots of, or they have some specific problems that make them possibly less suitable to use for children. And the main one is really that they are difficult to place. So you're probably going to need a local anaesthetic. You're probably going to need to do some drilling. You might need to isolate the tooth from moisture, which might mean putting like a little rubber dam, we call it, rubber raincoat around it. If you look at the pictures on the right of the screen, you need to use this blue jelly material, this acid etch, to roughen the tooth surface. You then need to place a bond layer. You can see that on the far right, the little yellow brush. That has to dry. Then you might have to cure that. Once you've done the bond layer, then you have to squirt in the composite, which then needs to be cured with a blue light again as well. So lots of separate steps. Makes it a little bit more time consuming, and children don't like this, can struggle with it. We know that it's toxic to pulp. To be fair, most dental materials are toxic to pulp, but that doesn't help, particularly if you've got a deep filling. And we know that composites, when they set, shrink. And when they shrink, they pull away from the walls of the tooth, and they leave little micro gaps, and then bacteria get into that. And one of the biggest reasons for failure with dental composites is then you get decay around the filling. The other problem that as well we're starting to realize nowadays is that there are enzymes in the dentine. The dentine is the part of the tooth structure that makes the bulk of it. And they will degrade the collagen, and collagen is something that's in the tooth structure. Apologies if you know all this, but just in case you don't. that helps, essentially makes up the structure of dentine. And composites don't inhibit enzymes that break it down, and that's a problem. So as part of this long-term work with Anne, we've sat down and we said, well, what do we want? And we actually asked children this. We said to children, what do you want? And I think the big thing is we want something that's easy to place. And so if we look at the image on the right-hand side, you can see a tooth that we've cut through. And so the top sort of shiny layer is the enamel, and then the main bulk of it is the dentine. We've shown that on the right-hand side. And on the left is a hole, is a filling, is some dental decay. So the top bit is the bit you really have to remove. Doesn't matter how clever you are, that bit's got to go. It's really infected, it's soft, it's mucky, that needs to go. Below that, you've got like a soft infected layer that maybe you could keep if you could somehow seal it in. Below that, you've got a softer but relatively infection-free remineralizable layer. And then below that, you've got dentine that you could probably leave behind. So ideally, if we want to make our filling easy to place, we want to reduce the need for drilling. And the way we could reduce the need for drilling is maybe just removing the beyond repair bit, because you could probably just scrape it out with an excellent. The second thing we need to do is to try and reduce the placement steps. So we've got this etch step, this bond step, this placing filling step, and then this light cure step. What can we do to eliminate some of those steps? We know with composites we have a problem with shrinkage and with recurrent caries. And we know that if we want to reduce the placement steps, we want to perhaps leave some decayed tooth behind or leave some demineralized tooth behind. So we need to reduce the risk of getting recurrent caries around that. And we want to also help the tooth to heal. So if we're leaving behind decayed dentine or demineralized dentine, can we put something in our filling material that's going to help it to repair? Finally, we want it to be biocompatible. So next slide, please, Anne. So that's what we did. And you can see a little video on the right-hand side just of the filling material being squirted into a tooth that we've cleaned with an excavator, not in a real human being. This was in the lab. We've well, more importantly, I think probably it's fair to say, Anne has created what we call a smart composite. So you can place it in one step. It self-etches and seals in one step. It mechanically stabilizes the weakened tooth, and Anne's going to talk about that some more in a minute. It's got some antibacterial action and also this anti-enzyme action, which I've already talked about. It remineralizes and it's non-toxic. And we believe, or we hope, that we can place this without local anesthetic, without drilling, without having to do this acid etch step, without having to do a bond step on a tooth that we haven't had to drill, or if you have to drill it, only a small amount, in less than 5 minutes. So that is our goal. That's what we're trying to test now if we've achieved. And I'm going to hand over now to Anne, who's going to dive into some of the chemistry of this particular material.
Thank you, Paul. If we're going to try and make a smart dental composite, what we first of all need to try and do is understand the chemistry behind the conventional dental composites. What these consist of is a liquid phase that consists largely of methacrylate monomers and a small amount of an initiator that's yellow and can absorb the blue light. We combine that with a high load of finely ground glass particles, and this gives us the paste that you saw in the last video. When you expose that paste to blue light, what happens? It sets off this chain reaction that pulls all the monomer molecules together. That gives us immediately a material that has got similar aesthetic and similar mechanical properties to the enamel and dentine that it's replacing. But as Paul said, pulling together those molecules can generate a gap. That gap's only a few microns, but that's enough for the bacteria to penetrate. In order to try and get around that, the first thing we've done is we've modified the R groups that are here on this chemical structure. If you make those methacrylate molecules larger, you've got a lower concentration of those methacrylate groups, and so you get less shrinkage when they polymerize. We also find that you can get it to polymerize more quickly. You can get higher levels of conversion. You can get deeper polymerization, and that allows us to put the restorative material in a single step, not do that layering and then curing each thin layer in turn. And if you have smaller number of those methacrylate groups, they're reactive and they can be a problem for the pulp cells. So we can reduce those concentration of those reactive groups. We've got better enhanced pulp cell compatibility. The other thing we've added in is some acid groups. What acid does is it etches the tooth structure, it etches the enamel and gives you a rough surface. What the liquid monomers do is they penetrate into there and they interlock with it. So that's how you can get the material to bond to the enamel. But if we're trying to bond to this carious dentine that's got some residual bacteria, what we really need is a way to try and stop those bacteria growing. And the first way we've done that is to add a new component called polylysine. Polylysine, it's got FDA approval as an antibacterial preservative, and it's got what's called GRAS status. That's generally recognized as safe. And so it can be used in our food. And this means it's going to be compatible with our pulp cells in the tooth. At the moment, we add this at concentrations of around about 0.1 grams per kilogram. So you consume quite a lot every day. What happens in the body is that degrades to lysine, which is an essential amino acid that we need in order to generate new tooth and bone structure. But in the composite, what we want it to do is to be released and provide some early killing of any bacteria that's been left behind during the tooth preparation procedure. But what we discovered is that the polylysine actually also absorbs water off the tooth surface and out of the tubule structure of the dentine. What this means is that the composite can penetrate into the tubule structure and it can interlock with it, it can provide retention, it can provide mechanical stabilization of that damaged tooth. It also gives effective sealing to prevent recurrent decay. What this next image shows you is how do bacteria grow in the presence of a composite disc. If we take about 1 million bacteria and put them into a well plate, leave them with nutrients, overnight they'll grow and you'll have about 1,000 more by the next day. So it'll grow from about 1 million to 1,000 million bacteria overnight. If you put a disc here, we've got a disc of Z250 as a commercial composite, put that in, it doesn't do anything to the bacterial growth. But if we put a disc that's got small amounts of the polylysine in, what we can see here is the bacterial number has gone down from 1 million to down below 100. That's a massive reduction in the bacterial growth. But the issue is, if we start off with a higher level of bacteria, then that level of polylysine can only inhibit growth. It can't actually kill. So we need more polylysine to actually kill if you've got a large number of bacteria originally in the tooth structure. So what we're really wanting to do is have, in addition to the polylysine action, we want to have effective sealing. If we can seal the cavity, what we know is bacteria decline in number. If they have no nutrients, they can't grow. They have no space, they can't develop biofilms, and they remain, and they will decrease in number underneath a restoration. So we need effective sealing. Paul says what the bacteria do is they produce acid that dissolves the mineral out of the tooth. And once the mineral has gone, we've got enzymes in our tooth that will cause degradation of the collagen the organic component of the tooth. So our own enzymes will degrade demineralized tooth structure. So that's why we have to remove that top surface tooth structure. But we want to be able to leave the tooth structure that's been affected by the bacteria, want to leave that behind and get it to remineralize. One way we can do that is to add monocalcium phosphate. What monocalcium phosphate has is it has those minerals that the tooth requires. They're the minerals that the bacteria caused to dissolve out of the tooth structure. So we want to put those back in. Monocalcium phosphate, like polylysine, it's got brass status, and it gives these minerals that we need not only for tooth, but also for bone repair. In food, we add this in concentrations of about 0.5 weight% to get bread to rise. It's a leavening agent in food. In the composite, though, we've discovered that it works with the polylysine. It reacts with the water that's been absorbed by the polylysine and helps the composite to really penetrate right deep down into that demineralized dentine structure. And then that sealing is going to help us to properly seal up the cavity, give the bacteria no space to grow and no nutrients. But what we're also doing is going to have this monocalcium phosphate provide us with the minerals to get that tooth to start repairing itself. But what we also discovered is that if you fracture a material that's got this monocalcium phosphate in, what happens over a short period of time is that crack that's forms actually fills up with minerals. So it gives a unique way for these materials to actually self-repair. If we look at a conventional material here, this is some CT images of a restoration, a commercial material put in into enamel and dentine. We can see here there's a gap down between the restoration, the enamel and the dentine. The smart composite, however, we can see it's adapted really well to this interface. It manages to seal up the interface of normal drilled enamel and dentine. But what is really unique is what this smart composite does when it's on the hand excavated tooth structure. It really penetrates a long way in, stabilizing it. What we've also seen is if we put a green fluorescent probe on, what this probe does is it fluoresces green if there's any enzyme activity. And what we see is that all the commercial materials we've looked at so far, there is some enzyme activity at the interface between the tooth and the restoration. But the smart composite, because it's effectively sealing, it's removing the water, there's no enzyme activity going on at that interface, or very little. So it's stopped the decay process. To look at whether the materials can remineralize, what we've done is we've taken a set material, placed it on top of demineralized dentine, and then taken it away after 6 months. What you can see if you put a commercial material onto the dentine and then remove it, you've got demineralized dentine, you've got all these open tubules. Whereas with smart composites, you get that surface is covered with the new minerals that the tooth needs to protect both the dentine and the pulp structure. So it looks as if the material's going to work well in the lab, but now the difficulty is, how do you get from the lab to the clinic? That's where I'm going to pass back to Paul.
Thank you very much, Anne. And yeah, I'll just finish by outlining this and actually where we're up to with our research and moving from the lab to the clinic. I mean, I could just leave it on this slide. You need money, probably a fair bit of it, and you also need industrial support. And I think it's important at this point to acknowledge the support of Schottlander, also DMG, who are manufacturing the composite for us, and actually, most importantly, I guess, NIHR, who funded via the Invention for Innovation scheme, really the work we needed to do to get us into a clinical setting. Next slide, Anne. So we had this material. We'd kind of gone through various cycles of optimization. In the end, we kind of felt, okay, we've got to make the leap now and see if we can get this to be a product that you can put on a shelf and purchase. And that it's not something either Anne and I had ever done. I think it's fair to say is an ongoing kind of marvelous complexity to try and get all that figured out. And you've got to start off by really putting together an evidence bundle, a technical file. You've got to decide how you want this to be classed and to kind of put that into some sort of perspective. I guess you could look at a weighing scale for a doctor to figure out how heavy you are as being a very simple classification. It's got to work properly, but you know, it doesn't have to do much else. Whereas you might think about a pacemaker, which you implant into a body and keeps you alive, as having a much more complex classification. So you have to work out how you want to classify your product, how complex, what potential it has to cause harm to the human body. You've got to make and package up a commercial trial batch to make sure that you can do that. You can do that repeatedly, that it's still the same material that you had in the lab. You've got to show that it's reproducible, that you can keep making it the same way. You've got to look at the shelf life, the long-term properties, its safety, its biocompatibility. Does it comply with standard dental materials tests? And then you have to move on to look at its efficacy, which is kind of where we're at right now. Next slide. And you need to prove that it's safe, firstly, to use in a human. Not really that it works, but that it's safe, that it's not going to cause an unexpected reaction, whether that be allergic or something else. And that means working with the Medicines and Health Regulatory Agency, who quite rightly are rigorous in their assessment of anything or using that's not been used in the human body before. You've then got to prove it works. Okay, so then you have to say, okay, fine, well, we can use this in a human being and it's not going to cause us a problem, but actually, does it do what we say it's going to do? Okay, and you have to work out how you're going to prove that and what standards you're going to measure against. And obviously, in our case, we're doing it with children, which clearly, quite rightly, again, adds extra levels of caution and difficulty. Once you've done that, then you take that evidence and you submit it to what's called a notified body. And that's essentially a company whose role is to sign off on what you've said, and you need to also put in with that your post-market clinical follow-up plan. So if you're going to use this, how are you going to make sure that nothing happens like 1 year later, 2 years later, 10 years later? So you've got to do all of that as well to get your CE mark. Okay, and obviously your CE mark is what means you can sell this in this country and abroad. And once you've done all of that, then obviously you can do your commercial production and marketing. So where are we up to? So we proved it safe just before COVID. Rather fortunately, we picked 7 children between 6 and 10 years of age who all needed 1 tooth extracting. So they were already going to have a general anaesthetic or have a procedure to have this tooth taken out. And we put the smart filling in this tooth for at least a month. And then when the tooth was taken out, we could take it to the lab and look at it. Okay, and a month was felt to be long enough to look for any sort of obvious problems. Results of that study, well, we showed we could put it in pretty quickly, less than 5 minutes, no needle, no drill, no pain. It lasted for the month and it lasted for the extraction, so that was positive. And when Anne chopped the teeth up in the lab, we could see that in small holes, we penetrated into the dentinal tubules. These are the sort of like these pores in the dentin of a tooth, and in the larger holes, we had sealed the degraded dentine. So that worked well, and we are now in the middle of our efficacy trial. COVID didn't help because it stopped everything, but actually it strengthened our case because there was a real, essentially children weren't treated during COVID, and it really has caused enormous problems and an enormous backlog, and obviously material like this, if it worked as we think it will work, would help with that. Brexit hasn't helped us really, plus changes in medical device regulations because everything's got more complicated. The goalposts have definitely moved. But we've started a trial funded by the British Research Council. It's what's called a phase 2 efficacy trial. We are placing the filling into one tooth into 125 patients, aged 5 to 9. We've recruited about 32 so far, and we're monitoring them over a year to see what happens. Has it failed? Has it not failed? Essentially, this is how we're going to prove whether or not it works. So that's all right now. After this, if this study works, then we can move on to CE marking. Once we get CE marking, then we can then go to a phase 3 trial, which is one you do in the real world with dentists working in practice, try and get more of a flavour for how it would work in different hands. You look at your real world evaluation, post-market follow-up. In the lab, Anne is working already at the next generation composites, building what we already know. We're looking to see if we can use it to cap pulps when the pulp is exposed, if we can use it for teeth that are affected by other problems, something called molar incisal hypomineralization, which is quite a common problem. And we're also even looking to see if we can repurpose it for bone repair. So we're kind of done. We'll do questions in a minute. I think before I go to my final slide, I just need to acknowledge all the people that have worked on this. I wasn't joking when I said it was a 15 to 20-year project, and a huge number of staff and PhD students and doctorate students and MSc people have been involved and continue to be involved. And these are not all the names, but they're most of them, I hope. But I think final slide, just to finish and to move into questions, I think whether or not our material works or not, I think it's fair to say that we don't really have, I think, the ideal filling material for children at the moment. That's not to say you shouldn't go to your dentist and take your children to the dentist and have what's there. And it's not to say that we can't deliver good quality work, but I think we could do better. We think our material, we think our smart composite, or actually, it's actually called a renewal MI now, but we keep it smart for this lecture, has got promise, but we have to complete our clinical trial before we can be sure about that. And I think for those of you who may not be familiar with the pathway to go from the laboratory to the clinic, I think just understand how difficult, expensive, and time-consuming that can be. Otherwise, thank you very much.
Thank you very much for the very interesting lecture. We all benefited from that, I'm sure. So we're going to move on to the Q&A now. And we've got a few questions up already, but if any of you in the chat watching had any questions, please kindly just enter it in the Q&A part on the bottom left, and we'll get to you once we get through the ones that have already been sent in. So firstly, first question asked was, will this material affect the resorption of the milk tooth?
Do you want to answer that one, Em?
Yes, so the resorption of the milk tooth, it's only really the root of the tooth that gets resorbed. Once the root has been resorbed, the crown comes off. We're only restoring the top enamel and dentine, so that will come off with when the tooth actually falls out. Mm-hmm. So only the root that resorbs, so that won't be affected. Okay,
thank you for that. So the next question is, are you aware of tiny dental implants being used to treat obesity in adults and children? And this is done by making eating more difficult.
I did have a look online about that. Yeah, my concerns with that are that you'd still be able to drink, and you'd still be able to drink things like Coca-Cola and things that got a lot of sugar, but then you wouldn't be able to clean your teeth. So yeah. I could see that could be a problem. I'm not too sure how many people would want their teeth locked together. I can't see too many people really wanting to go for that.
Yeah, it does seem quite odd. Well, the next question is on your opinions on stainless steel crowns in the NHS.
I think Paul used to take that.
Yeah, no, I'll pick up this one. So we do pre-formed metal crowns, probably the preferred term nowadays, all of the time. The smart composite isn't intended to replace those. It's an alternative. Preformed metal crowns are probably better for larger holes, and they're also quite good for uncooperative children using a technique called the Hall technique, where you just snap them on. Certainly, I work in a dental hospital. We do them all the time. I teach dentists. I teach them all the time. So yeah, they're widely used. They're widely used.
Okay, cool. Thank you. So, does fluoride as a child impact the development of teeth in your later adult years?
Yeah, and so I'll pick this one up as well. So one of the topics I teach here is around caries prevention and around fluoride. And when I started out years and years and years ago, we used to teach, or the thought was that fluoride consumption from a young age would make the adult teeth have shallower fissures and more rounded cusps, and that would make them perhaps structurally better at preventing caries. That's now thought to probably not be the case. And the reason that it's thought to not be the case is really because it was based on evidence from rats. And these rats were consuming fluoride in levels far in excess of anything that we could tolerate. Frankly, it would poison us. And they were rats. They weren't human beings. So I think nowadays the benefits of fluoride are thought to derive from topical use, whether that's in drinking water that you're drinking, what have you. And we don't really believe that you gain any benefits from consuming fluoride. Not that that's a problem, but I don't think we believe that it affects the anatomy of teeth anymore.
Thank you for that quite detailed answer. Okay, so some questions from the audience is it okay to steel over some decay on lower sixes with fissure sealant and monitor and restore it when older?
Yeah, so I'm going to take this one as a clinical one. And again, a topic I teach, the evidence nowadays is that sealing in caries, if done properly, is an effective way of stopping it progressing and is less damaging to the tooth. Now, there's lots of caveats around that. You have to be sure your patients understands what you're doing. You have to monitor this. You've got to be sure you put on a good sealant. You've got to check it. But I think that is a valid treatment choice. Yes.
Sure. So this is a question for you, Anne. How did you get to this stage in your career, combining your research with a clinical collaborator?
Well, I joined the Eastman Dental Institute after working for Shotlander Dental Company, and I was working on a lot of different things, but it wasn't until we had a meeting one morning and we met up with Paul that we really started getting together. But even after we got together, it took us a long time to find what it is that we could do together to work on. It takes a lot of time. It took a lot of working with different students, learning from the dental students, and as well as learning from the clinicians to find out what really was needed and how do we work well together. Takes time.
And I think to add on to that, it's sort of serendipity. So we met. At one point, this hospital arranged breakfast or coffee mornings to try and bring the clinical team and the research team together. And I was probably more looking for Anne than maybe Anne was looking for me. And I think we started out on one project, which was unrelated to dental caries. And then an opportunity came to apply for a small grant, something that was related to dental caries. And so then I got involved with that. And then one thing snowballed into another thing. So I think I sort of give a lecture that kind of answers that question. How did I get to where I was today? And I think if anyone presents a lovely structured, well, my target was this, and I did this, and I did that, that's absolute nonsense. I think it was a series of events, and it's all about capitalizing on opportunities that come, and some of them will pay off, and some of them won't pay off. I think we've both been lucky that this has been mutually beneficial to both of us. But I think we also both agree that we couldn't have got to this point without each other, because you need both sides.
That's great. So this is a question for you now, Paul. How did you combine your dental job with research? So that's essentially, you kind of answered that already, didn't you? Yeah. So you took the question right from me. So another question from the chat would be, what are the best fissure sealants, GIC or composites?
That's one for me. Actually, I mean, Anne, I guess I can flip this to you. In terms of bonding, which is better, resin-based or glass imomer? What
Well,
does the chemistry tell us?
bonding, you get better bonding, I believe,
To enamel, to be specific.
to the enamel. I think it's the resins. They have a stronger bond, and they are stronger materials. Whether that works clinically, there are a lot of not so good statistics saying that the fissure sealants don't necessarily last very long, but I think that might be because they're not placed properly. So
And yeah, and I think we would normally say if the child's cooperative enough, then probably a resin sealant is the way to go, because we would anticipate the bond would be stronger. That's not to say that you can't use glass ionomer sealants, but I would normally reserve those for cases where perhaps you have a less cooperative patient, and it's better than not doing anything at all.
Sure. So a general question was asked about the cost of these procedures.
Any procedure or?
The question, it just said how much has this process cost? So I think probably what you were talking about in your lecture.
Our process to get to where we are, gosh, I mean, I don't know.
probably had about 2 million in funding, plus similar amount maybe if you count the costs that all the students have paid to work with us. So probably about £3 million, that is.
That is significant. So,
Well, except it's probably not. It's probably cheap, really.
oh, is it?
Because a lot of the people that were on that acknowledgement list, a lot of them were, as Anne says, self-funded students who effectively working. Who would certainly there was a cost to somebody, but there was not a cost to the UK taxpayer for some of them. So I mean, I think one of the things I've discovered, because I hadn't realised, is research is expensive. Essentially. So and yeah, I think if you look at more medical interventions, then the cost just kind of goes up and up and up, because obviously people want to be secure and happy that what's being placed in their bodies is not going to cause them harm, which does happen from time to time.
Yeah.
But the other thing is that this is a very small amount of money in comparison with how much we spend every year in restoring teeth. So it's about 50 million just in having children's teeth extracted. I don't think about 3 billion every year just for tooth restoration. So it's a tiny amount in comparison.
So it's pretty worth it in terms of ratios of how much spent versus how much you'll probably save. Okay. So another question is, how do you fund the expenses for the whole process?
How do we fund?
So it's kind of similar to the last one, I suppose.
Oh, so who did we get funding from?
Yeah.
So there are a number of different sources. The sources we've got is from the government through the EPSRC is a research council. So the government gives money through research councils. We then have funding from the NIHR, which is the Health Authority, National Institute for Health Research. Then get money from overseas governments, will pay for students. We've also had money from industry. So industry has paid for a lot of the scale up and the manufacturing, producing, scale up of the products. So it's come from a lot of different sources at different stages.
Thank you for that. So do you think that there will be more AI work in dentistry in the future?
AI in dentistry. So at the minute, most of the work is around imaging. Can you look at an X-ray and determine if there's caries there or not? And that's about it. And I guess the field that maybe I'd be interested to see more in would be around treatment planning. I guess, but we're quite a way off that yet. And I think that's it's something we've got a small interest in, but I think we're quite a way off that yet. And I don't think anyone really fully understands what the impact of generative AI will be anyway. You know, depending on who you read, either the Singularity's coming, you know, next year, or it's going to be a damp squib, and you know, all it does is spread rumors, etc. So I guess we'll watch and wait.
Sure. Okay. So another question is, would the new composite be affordable and something that clinics would be financially able to invest in?
I answered that. The major thing that's going to cost money is people's time. So if you can reduce the time it takes to place a composite from 30 minutes down to 5, that's a massive saving. So it should be affordable. Also, you won't need to have all the other steps. You don't need the etchants, the primers, the adhesives, all of which are expensive. You just got a single material that is actually, it costs the same to make as a normal composite. So it should be a lot cheaper, because basically you don't have the clinician's time. Would you agree with that, Paul?
So, with regards to this material, do you believe that it could replace regular composite fillings in adolescents and adults as well?
At the minute, our sort of USP has been young children, and I think that's what we're testing it on. So I'm not sure we're in a position to fully answer that yet, Anne, would you say? What do you think?
Well, probably at the moment it's better for children, but it could also be very much for elderly patients in care homes, or in the third world, not third world, low middle income countries where they've got less access to dental facilities, those sort of situations. It's got a great deal more promise there. With adults, they can tolerate the longer time in the chair and the longer procedures.
Yeah, sure. So I think this has been mentioned briefly before, but could you just tell us again how long this whole process took?
I mean, we used to say 15 years, Anne, but it's got to be 20 now. Well, what do you
I suppose I started in about 2000. So that's getting up for 20, 24, 25 years now.
But I think it's important to note, Anne and I didn't sit down 20 years ago and say, we're going to develop this. You know, one thing leads to another thing, leads to another thing, leads to another thing. It took a while before we decided to commit to developing a material, because we just weren't sure, you know. So I suppose on the one hand, it's been a while. On the other hand, I don't necessarily want to paint the picture that to go from start to finish for anything takes that long. But certainly for us, it's been quite a long journey.
It's been made a lot worse by the COVID. That's really extended how long it's taken us to get through, and the changes in the regulations, which has made it more difficult to get products accepted. Yeah.
So when do you think that these new composites that have been being worked on will be ready for distribution in the market at like a larger scale?
I say. So at the moment, it'll probably be another year before we get through the phase 2 clinical trial. After that, it's going to the notified bodies, so it could take a year to get through the notified body. So the material's already been manufactured. We've got packaging lines and everything. So then we will need to do post-market clinical follow-up. It's whether the notified bodies then want even more data to prove that it really is going to last. So they may want a third clinical trial comparing with other products. So it probably, hopefully, maybe we might get there in about 2 years.
Hopefully.
Hopefully.
Right. And another question is that once you've completed this project, hopefully in a few years max, what's next? What do you have in mind? Like, do you run parallel projects between the dental and research labs?
So one of the things that we're trying to do is now look at whether we can place the material directly on the pulp. So at the moment, this material is not for putting on the pulp. So we're making new materials that could be used in that application. So then we'd be back again to go into the phase 1 clinical trial, phase 2 clinical trial, and starting all over again. So, and we've also looking into whether they can be used in bone for bone repair as well. So there are other applications.
That's interesting. Sure. So there haven't been any other questions. We've kind of exhausted them. So if anyone else has any last minute questions before we wrap up, that would be great. All right, so I don't think that there will be any more questions coming in. Oh, actually, there have been a few just now. Let me have a look at them. No, there haven't been any questions that have come in. So I'd like to say, first of all, thank you very much, Professor Paul and Professor Anne. This has been a very interesting lecture that I myself have learned a lot from. And I'm sure that we all have thoroughly enjoyed your expert insights on this topic. So that's going to be it for today. If you have any if you could provide us with your feedback about today's session, there will be a survey filled out that you can fill out. That will be emailed out to you guys, and it will be sent following today's event. That would be very greatly appreciated. So thank you to the audience as well for attending and making this lecture possible. And thanks yet again to Professor Paul and Ash and Anne. Thank you very much. Have a great evening, everyone. We'll be ending here.